to introduce Dr. Ned Sharpless, who became the 15th director of the National Cancer Institute last October, having previously served as the director of the University of North Carolina Lineberger Comprehensive Cancer Center since 2014. As an expert in the relationship between aging and disease, Dr. Sharpless has made numerous seminal contributions to the field, including identifying the molecular mechanisms of aging and senescence, the role of these drivers in cancer, and therapeutic opportunities to target them in aging and cancer. For example, I think you're all familiar with his team's elegant work that contributed to our understanding of the P16 tumor suppressor as an aging biomarker, and they also developed an important reporter mouse model to monitor P16 activation during neoplastic transformation and evaluate the age-promoting effects of environmental toxins. Dr. Sharpless received his undergraduate training at the University of North Carolina at Chapel Hill and medical degree at the UNC School of Medicine. He completed his internal medicine residency at the Massachusetts General Hospital and a fellowship in hematology oncology at the Dana-Farber Cancer Institute. After 2 years on the faculty at Harvard, he joined the faculty at UNC in 2002 and rose through the ranks, becoming the Wellcome Professor of Cancer Research in 2012 and then Director of the Comprehensive Cancer Center. Dr. Sharpless is a member of the Association of American Physicians as well as the American Society of Clinical Investigation, and he served on the ASCI Council from 2011 to 2014. He was an associate editor of Aging Cell and deputy editor of the Journal of Clinical Investigation. He has authored numerous original scientific papers and is an inventor of many patents. He's also co-founded 2 clinical stage biotech companies, G1 Therapeutics and HealthSpan Diagnostics. While being laser focused on developing his vision for the NCI over the past year, he has still managed to maintain his research program. We're really delighted to have him here with us today and look forward to his seminar entitled The Dynamic Interplay Between Cancer and Aging. So please join me in welcoming Dr. Sharpless.
Thank everyone for having me here today. I've had a really lively day and a lot of very interesting discussions, and I find these visits to cancer centers tremendously useful. I always hear about things that I didn't know were going on in the cancer research environment, and that is certainly been the case today. So thank you for a great visit and for teaching me a lot. I would also like to point out that I fully intend to leave some time at the end of today's talk for questions. I plan to finish early, so if there is some burning issue that you've always wanted to ask the NCI director, this is your chance. And it could be about the science that I'm going to show, or about the NCI vision that I'm going to show, or why your grant didn't get funded, or what, any of those things. I'm eager to take on. So, conflict of interest is very much the news these days. Out of abundance of caution, I show this disclosure information. The important word to note here is former. So to enter federal service, one has to divest of everything, and I did that. And but the Office of Government has actually asked me to continue to mention these conflicts for a year post-divestiture, and we're not quite there yet. And today I will talk briefly about some science related to these 2 companies, G1 Therapeutics and HealthSpan Diagnostics, and also maybe even mention Seminalytics, which is really the work of a company called Unity Biotechnology. So let's go. So almost two weeks from being a year in the new job. This is my wife and I being sworn in by Eric Harkin, the Deputy Director of HHS, October 17th of 2017. Is a very interesting thing to enter federal service from an academic job. The system is really different. You spend a few months just learning the acronyms and trying to meet the 3,000 FDEs that work at the NCI. And the federal process is different from the academic world. I should mention I started my career at the NIH. My first research job in medical school was actually on campus at the NIH, but I haven't been back since, other than to be on some study section or review board. And so while I've been an NCI watcher my entire career, there were still a lot of things about the job that have been surprising and interesting. One thing, I think probably the most striking first impression of the National Cancer Institute, here it comes an outsider, is just how big it is. I mean, it is an enormous organization. So these are 4 big programs within the NCI, and this is by no means all the programs we got. I mean, there's a lot. This slide would be much bigger if I tried to be comprehensive. But just to give one a sense of scale, you know, when you become NCI director, you are now involved in running a large research hospital on the building, on Building 10 on the campus. 30% of the patients in the clinical center are cancer patients, so we are by far the major user of this facility, which is, you know, the largest research hospital. There is no building, all the patients are in clinical trials. And the sort of maintenance and care and feeding of this is a problem and a really interesting topic. We have a huge national lab created by Congress in the sort of War on Cancer Act in the 1970s to give the NCI these special capabilities to move new technologies into usage very quickly through a government-operated, government-owned, contractor-operated, federally designated facility. It's based in Frederick, Maryland. And that is a very important set of capabilities for the NCI that we use for a number of activities. For example, the cancer genome atlas, the TCGA was administered through subcontracts by Leidos at Frederick, so it can be very, very useful to the NCI. The Comprehensive Cancer Center program is really, I would say, one of the most important things the NCI does. I'm standing here today at a great, you know, an exceptional, terrific cancer center. We're getting touchy about that. You know, I think I probably don't need to argue to this audience why it's important, but one thing that surprised me is that, you know, other national institutes of health don't really have a program like this. I mean, they have centers, but generally what they call a center is what we would call like PO1s. They're sort of, you know, multi-PI grants. But they don't have the sort of whole, you know, opus that the CCSG process is, both the good and the bad of that. But, you know, I would argue that the Cancer Centers program is really important for the NCI's ability to move ideas into, you know, practice, at least at tertiary care medical centers, and also to receive information from researchers in the external community about what's working in their, you know, in their cancer centers. And it's also a very important political part for the NCI. A lot of the communication to the both public, the advocacy, and to major, you know, the Congress is through Comprehensive Cancer Centers. So it's really useful, important part of the NCI's mission. And then lastly, I showed the National Clinical Trials Network to be illustrative of the several clinical trials networks the NCI runs. You know, these are major investments to prosecute clinical trials in cancer patients. And you know, a lot can be said about the Clinical Trials Network, but it remains vitally important to do certain kinds of studies, particularly large practice-changing studies in specific populations that are hard to fund through any other mechanism. And all of these things are, you know, very complicated entities on themselves, and there's a lot more to the NCI than what I've shown here. Maybe I'll say a word about the budget. The budget for the NCI has been good now. For a couple of years in a row, we've had this sort of nice, stable increases in funding on the order of $2 to $3 billion a year, so that in the last 4 years, we've had over $1 billion increase in our funding. The orange block shown here is the moonshot funding, so 21st Century Securities legislation provided $1.8 billion over 7 years for the National Cancer Institute. This year is the peak of the moonshot funding. We'll get $400 million for moonshot funding this year. Next year it will be $200 million. So that is a sort of cut to the budget, if you will. Although I would argue, you know, $1.8 billion is a good problem to have. But we do have to plan for it. Congress knew this was going to be an issue for us, and so they actually made the moonshot funding so-called any-year funding. It's not constrained by fiscal year the way the rest of our appropriation is. So that means that we can spend some of it now and pre-pay grants in advance, and that allows us to take some maneuvers so that the cut won't feel like a cut when it comes. I think that this doesn't happen by accident. You know, this kind of stable, predictable increase in funding has been really appreciated, and it reflects the broad bipartisan support for cancer research and biomedical research in general within the U.S. Congress at present. This is really, you know, it's an interesting time in Washington, as you've probably seen. But when I go down to visit the Congress, you know, I uniformly am greeted with, you know, how can we help you in your mission to try and do more against cancer? It's very popular. And I think the other thing that has to be said is there's a sense in Congress that it's a good investment, that the money spent on cancer research are leading to new therapies and new cures. And I think that's true. I mean, I think we've made a lot of progress in cancer over the last decade, and pace that exceeds any time in my career as an oncologist. And I think the Congress sees that. They see patients like Jimmy Carter, who they know well, and President Carter is a survivor of metastatic melanoma, which used to never happen. So they can tell that things have changed. So this success in cancer research sort of validates the investment. It also creates a bit of a problem that I'll speak to later, that the success has been uneven. So, if you have metastatic melanoma, your prognosis today is much better than it was 5 years ago. If you have metastatic pancreatic cancer, it's not much better than it was 5 years ago. So that uneven success causes some expectations and issues for the NCI that we have to talk about. I went and spent the first 6 months of the job trying to learn about the job. Went on this sort of learning and listening tour where I spoke to everyone I could speak to who had an opinion about the NCI. So I spoke to patients, and I spoke to scientists, and I spoke to federal officials, and I spoke to doctors and cancer center directors and advocates and every living former NCI director. I'm interested in some of the discussions. And I heard a lot of things that the NCI does really, really well and where the NCI is absolutely vital. And I also heard about things that the NCI could do better. And from that process, we sort of identified things where there were problems, but they weren't well suited for the National Cancer Institute. There were problems that we really couldn't fix. And spending resources futilely on such things, we wanted to stop. And then there were things where we were spending our resources on things that weren't really problems, and we wanted to stop that. And so the area we were really looking for, the sweet spot, were things that were problems where the NCI was well suited to help. Right? And those led to this sort of area, these 4 key focus areas. So one is, I would argue that perhaps the most important thing the National Cancer Institute does is training the next generation of cancer researchers. So developing the workforce and making sure that the next generation of scientists is well prepared for cancer research is really critical to our mission. We have a tremendous spend on cancer training, or researcher training. It's a big investment for the National Cancer Institute. And so I think it's also equally incumbent to us to ask, if we're going to spend that much money on training the workforce, are we spending it on the right things? Are we really getting results for those efforts? And if so, and how can we continue to do that? And so a question we've been asking ourselves a lot, and we've been asking the extramural scientists, is what skill set will a cancer researcher need 5 years or 10 years or 20 years from now? And will we train them for that? And we've heard a lot about that. I could make a talk on its own about sort of maybe the perceived holes in the workforce in the future. But I would say top of almost everybody's list, no matter who you are, is the sense that we don't have enough data scientists to address the data we're generating presently. And no cancer center director I've yet met has said, I have enough people in my cancer center doing, helping me manage our data. I think there's also some needs in the areas of immuno-oncology, but surprisingly, the need we've heard more voiced is around the clinical management of those patients than the basic immunology side. I think both are in need, but there's a pressing concern that we actually don't have the doctors to take care of some of the patients we're treating presently. And the list goes on. Basic science, that gets to this uneven progress thing. So it is not a fair statement to say that the NCI at any time in its history has ever been not interested in basic science. I mean, certainly, Harold Varmus was strongly committed to basic science. It is not a new thing for us to do. But the problem is, because of this uneven success, there is this sense that if the NCI merely just devoted more money to a specific disease or subtype, that uneven success would be rectified. That just more funding for this clinical trial in this area would be all it would take to make progress. And there are a number of people who would like the NCI to do that, would say spend a lot more money on this disease or this problem. And to me, that's always felt like skipping a step. The reason we made progress in certain cancers is because we had a scientific opportunity to make progress in those cancers. And without a biological understanding of these cancers, often that will lead to ineffective clinical trials. And in some ways, the worst thing we can do is fund very expensive trials that don't work. It's unfair to the patients, and it's a bad use of resources. And so I think that it has to be said that despite the fact that we've made a lot of progress in cancer, there are still some cancers that we really have to honestly admit we don't understand. And we need further basic biological understanding of it. So therefore, committing additional monies to basic sciences is important. Big data, I think, is a familiar topic to the University of Chicago, which has been an important part of the NCI's investment in data commons, for example. But this is still an area where we need a bit more work. And the thing I would like to see are data sets that are large, really large, that are aggregated so that we have radiology, histology, clinical outcomes, RNA, DNA, all that stuff in one place, and available to the research community with minimal barriers to usage. And of course we'll need some barriers to usage to make sure such data are safe and secure and privacy is protected to the extent possible. But this is an opportunity to make real progress for lots of patients in a very direct way. And I should also mention this is an area of great frustration in the advocacy community. Uniformly when I talk to cancer advocates, they're, why don't you know what happens to patients with body cancer at every cancer center in the country? Why do we read these reports of 25 patients with a disease and that gets published? And we need to know what happens to every patient with a certain diagnosis, and the way to do that is through intentional data aggregation. And clinical trials has been a problem area. A lot of things have happened in clinical trials and made it more difficult in any area of medicine. But I think the challenges, particularly in cancer, are great. And this is an area where I think the NCI needs to catalyze some innovative thinking, but also spend more money. And so we are trying to do both of those things. I will talk a little bit about these two for a moment, but I would like to say a few developments in these two areas before I move on. So I think the workforce development, probably some of the major highlights of the first year of an NCI are a big increase in our commitment to funding new R01s for early stage investigators. So we probably increased the number of ESIs by about 35% from the prior year, which is a big jump. And a new mechanism. So we've started funding some of the early stage investigators using a 7-year grant, the so-called R37, as opposed to a 5-year grant, the R01. And that's an experiment to see if extra funding or a longer period of funding helps early stage investigators. For basic science, I think the most important thing we can really do is fund investigator-initiated research. I don't think the NCI is smart enough to say this is the area, above all areas, where we need new progress most urgently, and we're going to devote our funds to that. We do use some RFAs, some directed funding for specific topics, but I think generally some of the most successful stuff in cancer is investigator-initiated, suggested by scientists in areas that the NCI haven't even counted as funding. So the best way to address basic science is to just put more money into the so-called RPG pool, the research program grant pool. And this year we've committed about, I would say it's on the order of $130 million for the RPG pool, which will be the biggest increase of the RPG pool since 2002, during which period the NCI budget doubled. Or during the period of the NIH budget doubling, rather, I'm sorry. So, that is, most of that money will go for extra, investigator-initiated extramural research, and we hope that will lead scientists like you guys to discover new stuff that will be good for cancer patients. Let me say a few words about big data. So, this is where we are now, and this is actually even an optimistic view. Really, if you look at what the NCI has today, we have a great genomic data set, the GDC, that is widely used, petabytes of data downloaded, hundreds of thousands of users. And then we have aspirations to have data in these other areas and to have them linked and usable. And this is where we would like to get to, as mentioned. So using a standard common data model, we can link and use these data together. And then we'd like to continue to add to these data sets. And, the NCI doesn't think it has figured this problem out 100%. We don't believe that we know exactly how to get there today. And so rather than invest tremendously in one Uber system, we are spending money on lots of different approaches to see what works and to try and get a better idea. One of the real problems for data aggregation is really this orange bubble. It's the clinical trials data. It's very hard to get the important data about a patient out of the free text of a doctor's note in some way that is usable for everyone. And that is a challenge that the biomedical research community will have to address. As mentioned, this is the most successful big data effort of the NCI at present. This is maintained here at the University of Chicago. Widely used data. This sort of started with the Cancer Genome Atlas. We are coming to the sort of end of the Cancer Genome Atlas project. The TCGA is sort of completing its last papers. And now we are starting to use that capacity, that sequencing infrastructure created as part of the TCGA to turn to new projects. And in general, those projects now include not just sequencing of the RNA and DNA of specific tumor, but also an analysis of the linkage with clinical data, for example. And this has been, I would argue, the TCGA is one of the most successful things the NCI has ever done. It's very, very important to cancer research. And it provides, it was our first sort of dipping our toe into the problem of big data in an area where we'd like to make a lot of progress in the future. So here are some of the investments we made in this area. All of these things are interesting topics unto themselves. Investing in data commons, working with the Department of Energy, upgrade changes and modernizing of the SEER program, which is a really large data set for the NCI. We have this problem of retrospective samples, right? So we have both how are we going to collect data going forward prospectively in trials in such a way that the data will be most usable and machine readable and aggregable. But even if we solve that problem, which we haven't, we have another problem, which is how do we learn from all the patients we've already treated? And how do we get information on the 20 years of patients that would be informative and useful to know about today? And so that problem kind of goes in two directions. There's the TCGA problem, where we've got a lot of sequencing data about the samples, but very little clinical annotation. And then there's the NCTN problem, where we have these big clinical trials with lots of clinical annotation and very little genomic information in some instances. And so we're trying to fill those problems in both directions. The TCGA, we're trying to annotate those samples to the extent possible, and the NCTN is this effort. So the Moonshot funded one program, or is funding one program, to go and collect the blocks retrospectively on several large NCTN trials and go back and sequence them. So to do the RNA and DNA to the extent possible, and just examples. And we will be announcing which of these studies we picked for that characterization very soon. This sort of started in a pilot version already by Lou Stout Center at the NCI, and so we've already been doing this on some NCTN trials, and that effort appears to be successful, and now we will augment significantly. And eventually all this data will be available. And there will be data where we have clinical outcomes, and we have response data, and we have genomic data, and then they'll be available to researchers to use, and ideally in a cloud-based format. So that is that problem I described, of going both ways for fulfilling missing data. And this clinical annotation problem, the TCGA problem is a big problem. We have a lot of samples in the genomic data commons that are not annotated, and we are now looking for solutions to try and fill that in to the extent possible for both the TCGA samples, target samples, which is the pediatric TCGA, and then a bunch of other clinical samples that we got where we have a little annotation. For example, Foundation Medicine gave us a number of sequencing results from their samples, some of which may be annotatable, depending on the level of consent of the patients at the time of the study. So a lot of work to both to fill in these missing big data. This is or make big data more useful in research meeting. This is a considerable expense. The Moonshot alone is committing something on the order of $50 million a year to filling in these holes and making these data sets available and usable to the research community. Let me now turn to a research topic. So I thought I'd take the rest of my time to talk briefly about cancer and aging. This is an area where I think I've been interested for a long time, and where I think the NCI could probably do more. We have known about the relationship between cancer and aging for a long time, but the basic research, the underpinnings of that, I think, is sort of in its infancy. It's the last few years, really, that some major advances, I believe, have been made to help explain and take advantage of the biological understanding of aging in terms of cancer research. So a couple things to say about cancer aging. For the first observation, this is just SEER data, which is registry data collected by the NCI, about the incidence of the most common cancers of humans. And the blue line here shows that there's sort of this peak around 60 years old, and that's true for basically every common cancer of humans. And they all seem to go down at advanced age, but it's not clear why this is. This could be an artifact of screening. We don't do mammography in older women, for example, or this could be biology. There may be something about people who survive cancer at this point that make them less likely to get it. It is a reproducible finding and sort of recapitulated by all cancer types. One of the reasons why cancer and aging are associated is now known. So processes that mutate DNA do so, some of them at least, in a time-dependent way. So this is a figure from Michael Stratton's work on tumor mutational signatures. Each signature is shown here. Across the top here are different kinds of cancer, and each signature reflects a pattern of mutations seen in the cancer that is associated presumably with a specific mutational process. So, for example, tobacco smoke has a specific signature, and reactive oxygen species may have a specific signature, etc. And you see, there's signature 1 here that's seen in kind of basically every kind of common cancer, and signature 5, which is seen in basically every kind of cancer. Signature 1 is the C to T transversions that's known to occur as DNA just sits around. Even if the cell doesn't divide, it undergoes spontaneous cytosine deamination and is sometimes misrepaired, and that causes mutations even in a static non-dividing cell. Signature 5, and is therefore a clock mutation. It reflects nothing other than the passage of time. It's effectively the half-life of DNA, if you will. Signature 5 is also a clock mutation. It accumulates with time. The cause of signature 5 is not known. But there are at least 2 distinct mutational processes that work on DNA in a time-dependent way. And some of the mutations they create are the mutations we know and love of RAS and p53 and the things that cause cancer. So even if your cell doesn't divide, even if you don't smoke cigarettes, even if you're exposed to any carcinogens, to no carcinogens whatsoever, there's some instability in DNA that leads to disrepair and carcinogenic transformation. So there's a lot of interesting things that one could talk about with cancer and aging. And, I alluded to one as sort of why the aging process causes cancer, and that is more complex than just the passage of time causing DNA mutations, and has to do with things like telomere shortening and heritable stem cell dysfunction and loss of cellular immunity and tumorigenic changes in the extracellular milieu. And similarly, there is a really interesting set of sort of clinical research now in this concept of resiliency, about why certain older patients with cancer get treated for their tumor and get cured of their cancer and recover marvelously, and then other older patients who get treated for the cancer may get cured of the cancer but don't recover whatsoever. And in some ways, the toxicity of the therapy exceeds the problems the cancer was causing them. And so why certain patients rebound from intensive therapy and others do not is not poorly understood. Is not well understood. And I think it's interesting biology. It also is a mechanism to test some aspects of aging in a clinical setting that might be interesting. But for today, I thought I'd talk mostly about this middle one, which is an area I found interesting as well, which is, to the extent that we understand the biology of aging, some aspects of cancer therapy, particularly the use of cytotoxic agents like adriamycin and cytoxan or platinum, as well as radiation, mimic aging. So some of the changes associated with physiologic, chronologic aging are not that different from what one sees with DNA imaging agents. And so this is a nice experimental paradigm that allows one to do human studies about the biology of aging, and I think has been interesting. So I have termed this sometimes iatrogenic aging. It's physician-induced aging in the treatment of cancer. And there's 2 kinds that I'd like to talk about briefly today. So one is this notion that cytotoxic chemotherapy induces this program called cellular senescence in certain replication-competent cells, and that those cells accumulate after cancer treatment or with aging, and that has adverse consequences for the host. And then the other is a different kind of aging, which is that this notion that as stem cells, particularly in hematopoietic system, are forced to proliferate, they become exhausted and have a change in their epigenetic state that makes them not good at producing blood and causes these biases away from lymphocytes and toward myeloid cells. And so this is sort of 2 different kinds of aging, both of which we think occur physiologically in humans as they age, and both are somewhat provoked by chemotherapy. And so I will talk briefly about both of them. So senescence is probably now familiar. I think it was pretty controversial for many years as to whether or not this actually occurred in humans. It was well established in cell culture, but the in vivo proof that senescent cells accumulate with aging has been challenging because the markers for senescence in vivo are not great. But I think now it's becoming reasonably well accepted that this is a real thing. These cells accumulate with aging and cause pathology. Senescent cells are identified by this enzymatic activity and excess activity of beta-galactosidase. They make certain other markers that I've shown here. They don't divide, so senescent cells are not proliferative. Probably the most useful in vivo marker of senescence is p16. So the tumor suppressor that prevents familial melanoma and pancreatic cancer is also an archetypal sort of mediator of senescence. And activation of this locus that includes p16 and ARF, the so-called ink ARF locus, the CDK2A locus, is a general feature of most senescent cells. Importantly, senescent cells make a bunch of cytokines, so they're thought to cause aging by promoting inflammation. And so these various hormones are shown here that are in general pro-inflammatory, and they have a few other markers that are interesting. And I would say, most of these SAS factors are in some part, to some degree, an NF-kappa B target. So the SAS is a persistent NF-kappa B activated state. And, I like to show, senescence was discovered by Leonard Hayflick in the 1950s when he was at the Wistar Institute trying to grow viruses in cells, and he didn't want to use HeLa cells for a variety of reasons, so he used primary fetal lung cells. And he noticed that they were always stopped growing in culture, and he called this phenomenon senescence. It didn't occur with cell lines. And he tried to publish this in 1961, and he sent his paper to the Journal of Experimental Medicine, and it was rejected because the largest fact to have come out of the tissue culture in the last 50 years is that cells inherently capable of multiplying will do so indefinitely. So meaning that if Dr. Hayflick had just known how to wash his glassware, all would have been well. And so he was accused of studying an in vitro artifact. His letter was signed by Peyton Rouse, who won the Nobel Prize in 1966 for his work on Rouse sarcoma virus. And I really like to tell this story in particular because if you think about it, the Rouse sarcoma virus, that led to the identification of SARC and the discovery of oncogenes. And so this is the first hint that oncogenes exist with sarcoma virus. And senescence, if you think about it, was really the first hint that tumor suppressor genes like p53 and RB and p16 exist. So basically, I like to point out that the oncogene guys have been dumping on the tumor suppressor guys since the field was invented. And that hasn't continued throughout my career until they put one of us tumor suppressor guys in charge of the NCI. So our moment has come. I will also point out this is a photo of me with Dr. Hayflick about a year ago in Santa Barbara. So he is very much alive and well, a very lively person, and goes to meetings and asks tough questions, and is a really interesting person to talk to. He, in fact, tells me that senescence is his third most important discovery. Number two was mycoplasma. He actually discovered mycoplasma, because when you're culturing primary cells, they get mycoplasma, right? So no secret why that happened. And he argues his most important contribution to science was the actual bi-double act. So he was pursued by the U.S. government for selling IMR90, or his WI38 cells, for about a decade. And eventually, he won his suit, or his argument with the U.S. government, and that led eventually to the idea of modern intellectual property. That you can take things you invent with NIH funds and commercialize them and make a company out of it. And that's not allowed in every country. So many European countries don't allow that. And that really provides a very different commercialization environment. So I think Dr. Hayflick may be right. That actually may be his greatest contribution to science, although I'm partial to senescence. The senescence theory of aging is this, right? And there are many theories of aging. There's a free radical theory of aging, there's DNA damage theory of aging, there's stem cell theory of aging, there are lots of theories of aging. But what I like about the senescence theory of aging is it actually makes predictions that we might test in our lifetime. And so, and it is this, is that senescent cells accumulate, and actually exponentially, so not just even at a linear rate, but at a very rapid rate with aging. And this is good for some things, like this limits cancer production, and GWAS data suggests this actually has a role in not getting atherosclerosis. And work from Judy Campese and others suggests that senescence may play a role in wound healing in some instances. But it also has some bad side effects, like the accumulation of those cells. They sit around, they're kind of in molecular jail, they don't divide, they make a bunch of toxic hormones. So they don't do what they're supposed to do. A senescent T cell can't divide and fight off infection the way a replication-competent T cell can. They occupy T cellular niches, so they crowd out cells that might be beneficial. At least in the case of atherosclerosis, some of the cells in the atherosclerotic plaque are senescent, we believe, and causing endothelial obstruction. And then they make the SAS, senescence-associated secret phenotype. And we think that's IL-1 beta and IL-6 and IL-8. Many of the hormones associated with getting a cold are made by senescent cells. So let me give three quick pieces of data for the senescence theory of aging. The first is if senescence caused aging, then people with different set points to activate senescence should age differently. And I think that turns out to be unequivocally true. We actually started out in my lab to do experiments to test this, and then we got scooped like nobody's ever been scooped before. It was like 20 GWAS studies on 2 million patients showed that the senescence machinery is associated with human disease in an unbiased genome-wide way that was almost breathtaking at the time. It sort of started in 2006 and probably finished in 2014. And so we kept noticing that a bunch of the senescence machinery genes kept coming up recurrently in GWAS studies. And so my lab downloaded, this is in 2012, but this sort of analysis has been reproduced subsequently by others, downloaded all the data, all the GWAS hits. And so each The chromosomes are ordered down here, and then the y-axis here is number of diseases that hit, and it's some GWAS to that interval of the genome. And we threw out all the things that weren't diseases. So we got rid of red hair and height and all that stuff. And so you can see there are 4 bins on chromosome 6 that are contiguous, so 800 kb region, that has lots and lots of diseases that map there, 20 plus diseases, and they're pretty much all things of autoimmunity. So type 1 diabetes and scleroderma and asthma and stuff like that. And then the next really significant, the dotted line here is statistical significance. The next significant hit is the heart locus, CDKN2A, and this is linked to basically almost every disease associated with aging, with one exception I'll talk about. And then these other significant hits, trust me, these things I haven't shown here are things that pretty much have to do with immunity inflammation, except for this one, which is 5p15, which is where TERT is, the catalytic enzyme telomerase. Telomerase is very important for cells not undergoing senescence. So if you think that regions of the genome should be associated with multiple diseases through a common set of pathological processes, this analysis would give you 2 candidates: senescence and immunity inflammation, and nothing else. There's no sirtuins, there's no IGF-1, there's none of the many things you might expect from an aging analysis that one will get from this analysis, one does not get, for reasons that I will not be able to explain. But another thing, if senescence were associated with aging, you ought to be able to measure it in people. You ought to say, who's got a lot of senescent cells at any given age, and that person ought to be physiologically older in some way. And that turns out, I won't, this is a long story, but I'll show just one piece of data, that this is a blood test my lab developed where you harvest T cells, because T cells undergo senescence, and you measure P16 in the T cells with aging. And basically, this is not to say that P16 goes up in every T cell with aging. What we think happens is, 1% of the T cells with aging have a 10,000-fold increase, and that's why it looks like this. But you see a very strong correlation between chronologic age of the donor and P16 expression in the T cells based on a log scale. So each 1 unit increase is a doubling of the T cell P16. This makes it very easy to measure. Any grad student can get this to work on the first try, as opposed to telomeres, which are a useful marker in interesting biology, but they change about 40% throughout the human lifespan. Very hard thing to technically measure. So we believe this is the most useful in vivo marker of senescence identified today. This marker is far from perfect and has problems. I can tell you about it if you're interested, but at least it exists. If you were going to try and clear senescent cells, and I'll come to that in a moment, you would like to have a pharmacokinetic marker response to your senescence clearing therapy. And we've used this assay in thousands of patients. We've looked to see if molecular age prior to therapy predicts toxicity, and it does in some instances. It correlates with progression and disease in some instances. It turns out to be an interesting story that I won't go into in detail today, but all the work is published related to this assay. And then lastly, and perhaps the most striking of the claims, is that if senescent cells cause aging, then getting rid of senescent cells ought to make an old organism young. This is paradigmatically different from what has gone on in aging therapy in my entire career of the aging researchers. Most studies were about preventing aging. Green tea. You drink green tea every day, and 30 years from now, you won't be as old as you would have been if you hadn't drank green tea. Terrible model from a farmer perspective, right? Who is going to do the randomized trial of green tea for 30 years, or exercise, or resveratrol, or whatever? So the prevention paradigm of aging is a non-starter from the point of view of pharmaceutical company. But the treatment paradigm of aging, where you take an old organism and make it young with a therapy, that's exciting to a pharma company, and it has really, I think, invigorated the senescence field a lot because of work like this. So this is a paper from Jan van Derzen's lab, who engineered mice so that he could kill their senescent cells when they were old. And he used a P16 promoter because that's really the only promoter that would work for this that we know about. And it drives a toxic gene that's activatable with a small molecule. So basically, you let the mice get old, you feed them this stuff, and all their senescent cells, or most of their senescent cells, die, and then you look and see what happens. And so these are littermates. This mouse and this mouse are exactly the same age. And you can tell this mouse is kyphotic, and has cataracts, and is losing body fat, it's a frail mouse. And this mouse is also over 2 years old and looks great. And if you put these things through physiologic tests, like how long can they hang on a rod, how well do the kidneys work, how do they do a water maze test, the senolytic mouse, the mouse that has been cleared of senescent cells, will perform better. This data was first published in a progeroid model, and most people, myself included, said, well, that's a progeroid mouse, that doesn't work. A mouse with accelerated aging was fake. So then it was reproduced in an physiologically aged model, and then we said, well, okay, but it's still a mouse model, it's one lab. And now it's been reproduced by other labs using completely different approaches, including genetic approaches and pharmacologic approaches. And I think at this point we have to consider it a true finding, at least in mice. Whether or not it worked in humans, I don't know. Whether or not senolysis in humans is even a good idea for a variety of reasons, I don't know. But the point is, we're going to find out. We will not be talking about the senescence theory of aging 50 years from now, whether or not we can prove it the way we talk about free radicals, right? We will either know it works or it doesn't sometime in our lifetime, which is why I'm excited about this field. Important to note, this also conveys a lifespan extension, so it doesn't just make the mouse feel better, they live longer. I'll just summarize the senescence portion of the talk by sort of saying what's the state of the art. The Montreal senescence meeting that I went to a few months ago was unlike any senescence meeting I've ever been to, or any age meeting I've ever been to in my career. One big difference was every single talk started with a disclosure slide. So all the scientists working in senescence now have equity in companies they started, or consulting gigs with big pharma companies. You know, for 20 years in the aging field, there's been no pharmaceutical industry interest in aging. A lot of interest in cancer. You know, I get called a lot about cancer ideas. Nobody ever called me about aging ideas until maybe the last 2 years when we sort of seen what it got done. So it's brought a lot of, you know, new thinking into the lab, into the topic. And as I mentioned, you know, it's led to rapid reproduction of some of the most striking findings. I was mentioning earlier today that, you know, it felt like kind of nothing happened in senescence research for about a decade. We just sort of were very descriptive. It was, you know, senescent cells make this and they accumulate here, and if you get rid of them, this happens. But, you know, in the last sort of 2 years, there have been a bunch of really basic biologic inquiry into the process of senescence that's very informative. For example, the finding that senescent cells really require active suppression of apoptosis. It's not just that they sort of stick around, they actually make genes so that they won't die, which, by the way, provides a vulnerability. All the senolytics are basically things that activate apoptosis for the most part. This is a really interesting biology after sort of a dearth of progress for a while. And now a bunch of human clinical trials. I mentioned some of the P16 trials. You know, those are sort of correlative trials. But more exciting, I think, are these therapeutic trials. So dasatinib quercetin is a senolytic regimen. And the idea at the Mayo Clinic is they give it to people with modest renal failure and see if you can make their kidneys work better. And then the Unity trial, UBX stands for the Unity drug, is to instill senolytics in arthritic knees. And see, the senescence, arthritic knees, we believe, are filled with senescent cells. And if you could kill all the senescent cells off, maybe you would feel better. I think, notice, you know, Unity or the senolytic companies could have done the trial to give a big infusion of these molecules and see if old people rise like Lazarus. They didn't do that. And the reason is, it could be toxic, right? I mean, nobody really knows what's going to happen. It doesn't seem to hurt the mice very much. But let's admit that the mice are a terrible model for human toxicology. So I think starting with the knee, a good idea, in my opinion. Let's talk to preventing therapy-induced aging, and this is a different story. You know, when we started this field, we hoped that it would also be related to senescence, and we did a bunch of work, and it turns out we think it's just a different way of aging. So part of aging is accumulating senescent cells. Part of aging is stem cells change their epigenetic fitting, and this is the latter. So hematopoietic stem cell aging is interesting. You know, these hematopoietic stem cells, they are really quiescent. They don't divide very much. They stay out of the cycle. You know, in contrast to what I was taught in medical school, that they are really sort of the spare tire. Most of the production of blood is done by progenitors on a day-to-day basis, and when every once in a while a stem cell divide creates new progenitors, or if there's an injury to the bone marrow, then they have to go on the cycle of coincid progenitors. But they're very protected cells that give you lifelong capacity to generate blood. Chemotherapy kills cycling hematopoietic progenitors. It doesn't really kill the deep quiescent stem cells, which is why we give people a big slug of toxic therapy and they regrow their bone marrow in about 3 weeks. So the ability to those stem cells to remain quiescent and to grow on demand is really important, we think, for the way we use cyclic chemotherapy in patients. Forced regeneration of hematopoietic stem cells does cause this thing called stem cell exhaustion. So you can show that there's a transplantable defect of such stem cells after serial chemotherapy. They just don't work as well. They don't make blood very well on a per cell basis, and they stop making lymphocytes. They have this myeloid skewing for whatever reason. And then lastly, we believe, based on work from Derek Rossi and Sean Morris and others, that the age state of hematopoietic stem cells induced by serial treatments of chemotherapy, or in animal models, something like 5FU, is molecularly similar to that induced by chronologic aging. So in other words, studying stem cell aging in these chemotherapy models is not irrelevant to chronologic aging research as well. And this work involves a trick, and this trick relies on modern understanding of the cell cycle. So I was taught the cell cycle worked like this. This is a model that still appears in textbooks and in review articles. It's pretty much wrong in every possible way. I mean, every prediction this model makes turns out to not be true for something in some regard. But like for example, CDK2, if a cell had to divide by activating CDK4 and then phosphorylating RB, or activating RB and then activating CDK2, if that was required for every proliferative event, then if you made a mouse that didn't have CDK2, that mouse would make it to the 2-cell stage and would die, right? And the CDK2 knockout mouse is fine. It has like no phenotype. So that's a problem for this model. So this model was thoroughly disproven by a bunch of mouse geneticists. It was based on work in yeast, C. elegans. But a bunch of mouse geneticists killed it off in about 2005 by knocking out all these genes and showing none of them had the predictive phenotypes. This is Peter Szynski, Phil Caldas, and Mario Martisid, and others. And the model that came about was a bit more nuanced, and it had some of the kinases working in some cells, and other of the kinases working in other cells, and some cells could only use one kinase, and some cells could use any kinase, and it became and some of the inhibitors inhibited one and not the other. It became a much more complicated model. But this had a very good implication for drug discovery. If you believe this is how the cell cycle worked, that a CDK2 inhibitor or a CDK4 inhibitor would be miserably toxic. It would make all not-dividing cells stop dividing. Who would want to do that? But if you believe this is how the cell cycle works, then maybe a CDK4 inhibitor wouldn't be so bad. And so this was met with a big, furious push in drug discovery to develop modern CDK inhibitors, some of which have been successful. So early on, when this sort of sea change in our understanding of cell cycle started to occur, I acquired some compound from Pfizer that was their CDK4 inhibitor. It's the drug that came to be known as palbociclib. At that time, it had a number. And we were like, if the cell cycle works this way, we asked this sort of naive question of what tissues in an adult mouse need CDK4 or CDK6 to proliferate? And the answer is most cells do not. Most dividing cells divide just happily. You can give palbociclib to mice for months and nothing bad happens. You can give it to humans for years. Not much bad happens. So but some cells are sensitive to CDK4/6 inhibitors. So the brown is a marker of proliferation. I think this is Ki67. And after treatment with palbociclib, you see all the brown cells go away in the beta cells. So beta cells need CDK4 to divide. You see all the brown cells go away in the intermediate lobe of the pituitary, which is something mice have that humans don't. So those cells need CDK4 to divide. You can see all the lymphocytes, either B cells or T cells, and lymph nodes or spleen, they all stop dividing pretty much. But every, vast majority is dividing cells. So the bone marrow cells that are dividing and the small bowel cells that are dividing pretty much divide just fine. They keep dividing on and on and on. And that's why the drug isn't very toxic. So the mice would get modest myelosuppression because the progenitors are dependent on CDK4. But other than that, they tolerated CDK4/6 inhibition pretty well. So that was good news for us because that meant that you could use CDK4 inhibitors therapeutically in humans. And you might use it in two ways. You might use it to prevent an unwanted cell from dividing, like a breast cancer cell. And you might also use it to protect a wanted cell from a cell cycle-specific insult. All the pharmaceutical effort at that time, at Novartis, at Pfizer, and at Lilly, was focused on number one here. How are we going to use these drugs to prevent cancer? And so we thought that was interesting and we were eager to work on that. But we didn't see anybody working on number two and decided we wanted to work on that because we thought that was cool too. And we spent a lot of time, here are some of the unwanted cells you might want to get rid of. I think it's much more than just cancer. You might want to get rid of, autoreactive T cells, or you might want to get rid of certain macrophage or monocyte cells that are deleterious for some reason. But here are the cells you might want to protect. And I'm going to talk about this one today, not with some engineers. But I would mention the kidney thing is fascinating. Kidneys are deeply quiescent organ. They don't really divide much until they're injured. And then the renal epithelial, like 50% of the cells can be dividing in S phase within 24 hours. So throughout life, they maintain this really amazing facultative response to injury, which we know as doctors, right? You give them the platinum, their creatinine goes up for 3 days, and then it goes down. They kill all the epithelium, and then it regress. It's really marvelous. And that entry into the cell cycle is totally dependent on CDK4/6. So I think there's the ability to prevent certain renal insults that are cell cycle dependent. We haven't done much work on, but I think it's a story that would be worth pursuing at some future date. We couldn't get palbociclib or abemaciclib or ribociclib for use in these studies, in human trials. We could get stuff to use in mice, but if we wanted to do human studies of protecting hematopoietic progenitors, it turned out we eventually had to make our own drugs. So we started a company and got an SBIR and did a bunch of stuff in drug discovery, and it took like 7 years. And I won't show any of that. And what emerged out of that was this drug called trilaciclib, which is also nicknamed G1T28 or G1 Therapeutics 28. It looks like ATP, so this part of the molecule is sort of the ATP mimetic, is a classical type 1 kinase inhibitor. It does what it's supposed to do in RB-competent cells. So if a cell is RB and the CDK4 dependent, it makes it stop dividing. If a cell is RB null, it doesn't do anything because CDK4 phosphorylates RB, and if RB is gone, the effect of the drug is non-existent. And this drug was, as opposed to all the other CDK4 inhibitors that were designed for oral use for cancer. Think about it, you want a drug that you can take as a pill, that has a very long half-life, so you take the pill once a day. This drug was designed to protect the bone marrow in transient periods. And so it was IV, because if you're going to protect someone's bone marrow from cytotoxic therapy, you would rather they didn't throw out the pill. And we also designed it to be short half-life, so that when we stop the infusion, the bone marrow would not divide for a while, and at some point it would wake up and start dividing again, so that it wouldn't cause indirect secondary myelosuppression. Here's a summary of the existing CDK4/6 inhibitors that I'm aware of that are in clinical trials. Trilaciclib, I just talked about. It's an IV, highly potent 4/6 inhibitor. Palbo and Ribo and abemaciclib are FDA approved for hormone receptor-positive breast cancer. These two, and actually trilaciclib, I think are somewhat similar. This one, abemaciclib, is quite different. It's a staurosporin derivative. It's a planar molecule. It's a much dirtier compound. One thing we discovered in doing this work, when we screened some 200 compounds in our series, is that if you want to protect bone marrow, you need a really good 4/6 inhibitor, and it can't inhibit CDK2 at all, right? So it has to be highly selective. That was not something we thought would be the case going into the studies, but for reasons I won't go into, I think it makes perfect sense. But so you notice these compounds are all 500 to 1,000-fold selective for 4 and 6 over 2, but abemaciclib is not. It is a much dirtier compound that hits CDK2 even at achievable doses. And then liriciclib is the oral G1 drug, which is very, in some ways similar to palbociclib, highly potent and selective for CDK2, but an oral compound. So let me briefly remind people of hierarchy of hematopoiesis. They're these stem cells that I already talked about. They don't divide very much. They give rise to these progenitors. They divide a lot, which then give rise to committed progenitors. The multipotent progenitors are easily identified by immunophenotype. So the cell we use the most is a so-called LSK cell, lineage negative, SCA positive, K positive. That's a great pharmacodynamic assay. So those compounds that we screened, we would then give to mice and harvest the LSK cells and see if they stop dividing, because we knew those cells to be CDK4 dependent. And that was how we screened drugs for pharmacology and pharmacodynamic effect. And we were able to rattle through a lot of compounds using that assay. I will go ahead, I won't show the data, but pretty much all of these cells from kind of common myeloid progenitor on, and this is not drawn to scale, it's like 20 proliferative events from that cell to that cell. But all of these cells on this side are pretty much CDK4/6 refractory, whereas these cells are CDK4/6 dependent, as are all the lymphocytes, interestingly. We still don't really understand what that is. But T cells and B cells are very sensitive at basically every stage. So as progenitors and then as differentiated effector cells. Here's some graphing of that data. So this is using trilaciclib, where we've done it with palbociclib and other compounds. Hematopoietic stem cells and LSK cells and T cells, as mentioned, are very sensitive to these drugs, whereas erythroid progenitors not so much. If you give a single dose of the drug, the hematopoietic stem cells, or this could be LSK cells, you'd see the same result, stop dividing for about 24 hours, and then they wake up when the drug goes away. And so this was the idea, right, is during this period when the stem cells and hematopoietic progenitors have gone to sleep, you could give the platinum, right? That would be the time to give the DNA-damaging agent that would kill the cancer. You see right away there's a problem. The cancer has to keep dividing, right? So you have to have the cancer be refractory or not need CDK4 for proliferation, but the bone marrow is protectable during that period. So what kind of cell cycle insult? Well, it's been known since the 1960s that DNA-damaging agents kill in a cell cycle dependent manner. I mean, this is talked to like grad students and radiation oncologists the first day they start. So early on in G1, quite resistant, right at G1 S reversal, quite sensitive. This is fascinating biology. Even though we've known about this for 50 years, I think we still don't fully understand it on a molecular basis. But it's well established, this relationship. And importantly, anything you do that will extend G1 will prolong that period of radioresistance. So the idea here is that just by extending G1 with a CDK4 inhibitor, you allow damaged cells to recover before they divide and die. And it works. So here's a mouse experiment using trilaciclib, although, as I said, we had very similar results with palbociclib. Here are untreated mice and their blood counts. Here are animals that get a single dose of 5-FU. These are not all at the same time point, so the kinetics of myelosuppression are different for different lineage elements. So this is sort of the nadir counts of each blood element. But here's 5-FU in white blood cells, neutrophils, and lymphocytes. the red blood cells and the platelets, 5-FU makes all of them go down. And if at the same time as the dose of 5-FU, you give a single dose of the CDK4/6 inhibitor, you rescue neutropenia and lymphopenia and anemia and thrombocytopenia. So one dose at the time of chemotherapy provides quadrillion protection. Hematologic toxicity is really important. It is the dose-limiting toxicity of most cytotoxic chemotherapies. It is the reason patients have their doses delayed and gets reduced dose intensity. It causes a lot of morbidity, including febrile neutropenia, which is expensive and lethal. And so if one could do something about it, one might make these good old drugs like etoposide and carboplatin and erythromycin better drugs. If they worked but would cause myelosuppression, you might like to use them better. So all the studies of the G1 process look like this. There is some chemotherapy drug given in a cyclic way, and at the same time as the chemotherapy, trilaciclib is given, IV, on the day of the chemotherapy. So etoposide platinum is a 3-day regimen. So you come in and you get, while you're getting your rehydration through chemotherapy, you get trilaciclib before the chemotherapy on days 1, 2, and 3. And you repeat that every 21 days. Some of these studies are for first-line small cell lung cancer. Some of these studies are for first-line small cell lung cancer with etoposide platinum plus PD-L1 inhibitor, a genetic study to see if by protecting the lymphocytes you can make an immunotherapy agent work better. And one of these studies is in breast cancer, and triple-negative breast cancer. And the trials are all in cancers that are thought to be RB deficient. So tumors where the cancer will continue to proliferate because it's functionally RB deficient, whereas the chemotherapy, whereas the CDK4 inhibitor will block the bone marrow proliferation. And so here are the first human clinical data presented about 2 months ago. This is in a first-line small cell trial. EP is etoposide platinum. It's a randomized phase 2 placebo-controlled trial. Here's platinum plus etoposide platinum trilaciclib, and you can see all of these hematologic endpoints are substantially and significantly protected by the co-administration of trilaciclib. So things like grade 4 neutropenia in the first cycle are reduced 90%, and dose delays are reduced 40%. And there's a favorable trend to other indicators, including prevention of this problem. This is, by the way, been presented in the form of a press release. This has to be done because it's a material event, so a publicly traded company has to disclose this as currently as possible to comply with SEC requirements. The real data get presented at ESMO in about 2 weeks. So, in summary, cytotoxic chemotherapy cures some cancers, but untowardly induces the accumulation of senescent cells that drive aspects of aging. Senescent cells appear to drive some of the long-term toxicity of cancer chemotherapy. Cytotoxic chemotherapy induces stem cell exhaustion in part by depleting hematopoietic progenitors and then forcing the stem cells to have to regrow your bone marrow. And pharmacological quiescence, the CDK4/6 trick I talked about of hematopoietic progenitor cells at the time of cytotoxic chemotherapy can prevent some aspects of stem cell aging. In the interest of time, I didn't show that data, but we've done a bunch of transplant experiments that provide support for that last conclusion. And here are the people who helped with the work, some of them back in the University of North Carolina, and some collaborators at other institutions. And I'd be happy to take questions about the NCI or about the science I presented. And thank you for inviting me. Oh, I didn't leave as much time as I expected, but I tried.
So, in your last study, the drug that you used was an ATP inhibitor, and so you could imagine that the cancer cells would actually develop a resistance anyway, which would be very effective. So, can you imagine doing it carried out of chemotherapy?
Yeah, I had this, as we published a paper like this, and the reviewers just hated it. But, we were making the claim that you could kind of use a CDK4/6 inhibitor in every cancer, right? Because think about it. At first, you could give it to them, and their cancer would stop growing, and then it would be an antineoplastic, and that would be great. And then at some point, it would become resistant to the drug, and then you could use the protective bone marrow to use that inhibitor. So we had sort of this like group 1 and group 2 indication. One big problem with that has nothing to do with science and everything to do with commercial development, which is that you don't want the same molecule for the same purpose for 2 different applications, because then how do you price it? So this is the Lucinta Avastin problem. And while that sounds like a great idea to scientists, that's a terrible idea to a CEO of a biotech. So there are 2 different molecules going
But
forward.
related to that, then, a lot of drugs now are being developed as allosteric inhibitors as opposed to ATP. Have you thought about that as an alternative?
Well, by the way, I should mention I am completely divorced from G1 Therapeutics. Nothing I know about what they're doing is anything other than public information. I at one point was on the board and chaired the SAB, but those relationships have all been severed. So I don't know anything that's not available in the newspaper. But I believe that in the time I was affiliated with the drug discovery efforts, we spent a lot of time looking for a whole bunch of different CDK inhibitors by a whole bunch of different approaches, and we never really found anything that looked like a good health therapy inhibitor. And I think that's probably a general phenomenon, because I'm not aware, there have been many medchem efforts around CDK4/6, CDK2, and CDK9, and CDK7 now even, and to my knowledge, none of them are health therapy inhibitors. So that just may be, as you know, those things are flaky to find. But when you find them, they can be really great drugs. But we didn't find such a thing. We would really like to have a compound that distinguishes CDK4 from CDK6. We were never able to find that either. But I think people are working on it.
So going back to the P16 studies, I was curious whether, I think it was good to see whether P16 in any way regulates mitochondrial function or also production and means to reconciling the P16 aging theory with the old drugs, the steroid agent.
There's certainly been a lot of work on that topic in senescence. So a lot of people have focused on the redox state of senescent cells. I would describe that literature as, a lot of it hasn't given a clear signal as to whether or not redox is positive or correlative. I think it is generally true that some senescent cells at least seem to have activation of redox pathways. Most of that phenotype is completely recapitulable if you knock out P16 and the cell still senesces in a P53-dependent way. So, you can get something like that in cells that will go senescent, that will senesce in a P16-dependent way, which is not every cell. But in some cells, at least, you can get senescence that looks every bit the same, whether or not it has P16. So I think, we, in my lab, spent, years trying to find things that P16 did other than inhibit CDK4 and CDK6, because it didn't make, there are 4 ink molecules, 15, 16, 18, 19, but only one of them is the tumor suppressor gene. So we thought it must have some magic special property. It must do something else other than inhibit the cell cycle. And we could never find it. And we did, RNA expression profiling and mass spec and all kinds of, unbiased experiments. And basically, we always got things that have to do with CDK4, CDK6, or the downstream targets.
I know that you have a very tight connection, so I want to thank